All right, did everybody get my notice that I posted the slides for T-cell development? I forgot to post them yesterday, so. All right, this is a review of the lecture we had last Thursday. So it's a great review. You can go through it, and I prefer not to go through it step by step, so I did it pretty quickly on Thursday, but seriously, it'll just help crystallize what we went over on Thursday. So use this as your study guide. I think it'd be quite helpful. All right, this is our final data analysis for this topic. And what we want to look again, remember ZAP70, which is AS, this is plus minus AS, which means that it is sensitive to the inhibitor. And again, the inhibitor is a long name that we need not worry about because I certainly can't remember it. And in this case, we're going to activate the T cells again by using anti-CD3 and anti-IgG, which cross-links the original antibody to get cross-linking to get activation. And so what we're going to look at are two different classes of T helper cells. One is T helper 1. And one is T helper 2. Now, at this point, you don't know the difference between T helper 1 and T helper 2. Just to alert you, we're going to learn all this, probably not before the second exam, but there's T helper 1, there's T helper 2, there's T helper 9, there's T helper 22, there's T helper 17, there's regulatory T cells, there's also follicular helper T cells. There's a whole long list of different T cell types. So don't worry about them because you don't know what they are yet. The only reason I'm showing you T helper 1 versus T helper 2, cell lines of T helper 1 we know produce gamma interferon as their iconic cytokine, whereas T helper 2 is a cell line of T cells which as their iconic cytokine produce interleukin 4. That's all you need to know to understand what's going on with these data sets. So two different types of T helper cells. And when we stimulate them with, again, the antibodies which allow cross-linking, we look at whether or not we have CD4, which is the y-axis, and gamma interferon, which is the x-axis, and double positive means that this particular cell is double positive for both these characteristics. So in the upper right, this is a CD4 cell, which also is able to synthesize gamma interferon. By the way, we permeabilize these cells, so the antibody to gamma interferon enters the T cell and is able to bind to it and fluoresce as a result. So that's our double positive. And then as we add inhibitors to our to the sensitive ZAP70, you notice that the upper quadrant, right quadrant disappears, just as you would predict. However, the final row here, or column here, we're going to artificially stimulate, and I want you to think about this, with two molecules, PMA, 4-bomyristate acetate, which mimics protein kinase C, which mimics diacylglycerol, or activates protein kinase C, which mimics diacylglycerol. So we're going to artificially, if you will, substitute for diacylglycerol. We're going to add PMA. And then we're going to substitute for IP3 pharmacologically by adding a molecule called an ionophore. Okay? And ionophore allows calcium to come in instead of IP3 being the signal for calcium to come in and be released from the ER. It's called an ionophore. PMA substitutes again for diacylglycerol. And remember, we get IP3 and diacylglycerol by activating phospholipase C, which gives us, which works on phosphatidylinositol, which gives us diacylglycerol and IP3. Now I want you to talk and think about this. why was this last column done? Why was this experiment done? And what did the data tell you about the cells that are involved in these experimentations? So there's a reason this particular experimental procedure was done. I want you to analyze that and think about it. That's your mission right now. Think about why that last column was done and what it tells us. Look at the data. That's a clue. Look at the data. All right, let me help you direct this. The whole point of our discussion about activating T cells was to get to transcription factors which eventually lead to the transcription and translation of cytokines. So first of all, this data absolutely shows that's the case. If we stimulate the cells, whether T helper 1 or T helper 2, we get the iconic cytokine being produced, presumably by the messengers or the transcription factors that we're inducing, like NF-κT-C and nuclear factor kappa B and the MAP kinase, what have you, and we end up with double positive upper right quadrant in all cases. If we add the inhibitor, of course, the upper right quadrant is compromised because ZAP70 is no longer effective, and then that begs or raises the question I gave to you: what's the point of this final column? Compare this to this. Here's the inhibitory sensitive ZAP70, and this is the non-sensitive inhibitor, non-sensitive ZAP70. Well, what did the ionophore do? As well as the 4-bomurostate acetate. That signaled what? Upstream or downstream from ZAP70? Upstream or downstream? Downstream, right? So therefore, even though we have a defective ZAP70, everything downstream is still intact and working because we substituted for the functions of ZAP70 by giving it an artificial diacylglycerol, if you will, and giving it an artificial IP3, PMA, again, an ionophore. And can these inhibitory sensitive ZAP70 cells still produce cytokine if we bypass the ZAP70 roadblock? Can they still produce cytokines? Yes. So why, as an experimenter, would you do that? You want to make sure that in this process, that messing around the ZAP70 did not interfere with anything downstream to that particular molecule. And this shows you everything downstream is probably intact and works perfectly fine. Because if we give the downstream molecules, again, substitute for DAG to substitute for IP3, yes, we will get gamma interferon for our T helper 1 cells, or IL-4 for our T helper 2 cells. So everything works perfectly. You can compare this upper right to this upper right. There is no IL-4 in this case, and there's copious amounts of IL-4 in this situation. Here, there's no gamma interferon, and there's lots of gamma interferon in this case, in the upper right. Yes?
So it's
Allegra.
because the calcium activated pathway still works?
That's exactly right. Because we gave calcium, which substitutes for the need for IP3. Because remember, IP3, all it's really doing is leading to the release of calcium. from the ER, which leads to even more release of calcium from the membrane through the crack channels. So same type of thing. Okay. So this is a great experiment because it shows us two things. First of all, that ZAP70 will lead eventually to transcription factors, presumably, which give us the end product we're interested in, which are the cytokines. That's what I said all along. So I didn't lie to you. Promise. And number two, if we interfere with ZAP70, we know that the downstream effects work, and they're certainly, in this case, most likely due to diacylglycerol and IP3, treating the calcium and the accompanying effects. So this is a really nice experiment, which I truly like to show to classes. Okay. Now, if you activate a system, you have to have some way, evolutionarily speaking, have developed to come about to deactivate it, because a system which is permanently activated will run amok and be quite injurious to the host. So you have to have some way of deactivating it. And the opposite of a kinase is a phosphatase. And it turns out the only way we're going to look at deactivating our signal, in many ways you can deactivate a signal in our story. And obviously I'm not going to go through all these different methods. We're just really going to look at phosphatases, which is the main means by which this is carried out. And phosphatases do just as their name is defined as. They remove a phosphate group, and these are tyrosine phosphatases. So they remove phosphate groups from tyrosines which have been phosphorylated. Now, how do you activate a phosphatase? Well, it's a very good question. So let's have a story here. Now, remember ITAMs? Everyone remember ITAMs? Now, ITAMs have a tyrosine which are surrounded by a set of conserved amino acids which are recognized by tyrosine kinases, such as SAR kinases, and then can phosphorylate that tyrosine. Okay? And again, it's not any tyrosine the SAR kinase will phosphorylate. It's a tyrosine surrounded by a conserved set of amino acids, leucine, isoleucine, valine, etc. Now, those are called activation motifs, ITAMs. But there's also the mirror image of our ITAM. This is really good for understanding cell biology, called ITIMs. These are inhibitory moderating regions in a particular protein. And they contain tyrosines as well, with a different set of conserved amino acids. Okay. And this particular ITIM, if their tyrosines are phosphorylated, will bind to phosphatase and activate it. Okay. Now, the ITIM, just like the ITAM, is phosphorylated by a SARC kinase. So now we have a SARC kinase. Remember, when the T cell receptor binds to the MHC and the processed peptide, that leads to, remember, cross-linking. And therefore, our SARC kinases are dephosphorylated by CD45, and then the SARC kinase phosphorylates tyrosines at the ITAMs, to which combine ZAP70, et cetera, Well, it turns out ITIMs may also be phosphorylated, and then that begs the question that if they can bind there for a phosphatase which becomes activated, why don't these phosphatases just counteract the effect of the tyrosine kinases and you get no effect? You know what I'm saying? So if we have the tyrosine kinases activated by binding to the ITAMs, at the same time we have an ITIM which is phosphorylated and binding a phosphatases which is becoming active, why doesn't it just counteract effects of the tyrosine kinases? Get what I'm saying? They have both the kinase and the phosphatase activated simultaneously. They should just balance each other out, and the net effect of +1 plus a -1 is equal to what? What's a +1 plus a -1 equal to what? 0. So we shouldn't have any effect whatsoever. Well, we know that's not the case. So why don't we have 0 effect? Well, anyone can think of that? Why don't you have zero effect? Well, first of all, the go-to answer in immunology is no one knows for sure. Okay, so if you say that on an exam, I'll give you a couple of points because that's absolutely correct. But let's go beyond a 20% out of 100% for a question. All right, and let's say no one knows for sure, which is really true. Does anyone think of a reason why you wouldn't get -1 plus 1 equaling 0? Alana, since I was standing in front of you, maybe my brain went into yours, so go ahead.
Because they're
And maybe yours went into mine. Go ahead.
they're not exactly equal, so sometimes there's more kinases, sometimes there's more phosphorus.
Yeah, that's right. So that's right. I mean, it seems like a straightforward, simple answer, but it's absolutely right. When you have the TCR binding to the MHC plus processed peptide, that's going to favor the tyrosine kinases, but the phosphatases will also be activated. But what happens when the level of antigen starts to decrease? After a while, you're going to start destroying the antigen, and levels of antigen will start to decrease. So if you start to decrease the levels of antigen, you're going to shuttle more to the phosphatase, probably, part of this equation, and end up with deactivation. That's one possibility. Another possibility is that the molecules which have ITINs on their cytoplasmic domains may not get expressed until later on in an immune response. So you have the TCR binding to the APC, recognizing MHC and processed peptide, and that activates a tyrosine kinase cascade, blah, 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 et cetera, et cetera. Then later, as the immune response starts to wane because antigen levels start to decrease, there's a timing element here. The T cell expresses a breaking molecule that's not expressed until later. And I'll give you an example of one of these breaking molecules, PD1. I've typed in here, PD1, which stands for Program Death 1. And PD1 is expressed later after the T cell's been activated for a while. So it's not expressed on naive T cells, but only on activated T cells and after they've been activated for a while. Because you wouldn't apply a brake to a car unless you first turn the ignition on the car and started to drive, right? So this is the same type of thing. PD1's a brake. And you're not going to have it expressed until after you get behind that steering wheel, either use a key to turn your ignition or a push button, whatever kind of car you have, and you start the car, and then you pull out of your driveway. Well, you should use a brake then as well. And you get what I'm saying with my contorted, hackneyed metaphor here. And so PD1 gets upregulated later in the immune response, and it has lots of ITIMs on its cytoplasmic domain. Remember, ITIMs have tyrosine and conserved amino acids around it, isoleucine, etc. You don't have to know what those are. And that will then attract a phosphatase, and the classic one is SHP, and there's another one called SHIP, SHP and SHIP, SHIP. And once that's attracted to the phosphorylated region of ITIM, that kinase is then, phosphatase rather, is activated, and then it can dephosphorylate tyrosines, which have been phosphorylated on ITAMs, and lead to a deactivation of the whole process. So that's how these brakes work, by the way. The PD1, there's other braking molecules which may or may not work through ITIMs, but PD1 certainly does, and that's how you may deactivate the system. Okay? Any questions about that? Yeah.
So you said SHP helps activate PD1 and then PD1...
No, PD1 gets upregulated first because it's a process, a delayed process. And then once PD1 is upregulated, you have these ITIMs because PD1, I didn't tell you this, binds to a a receptor on APCs. Okay, it's called PD, or ligand rather, it's called PDL1, program deaf ligand 1. And that causes cross-linking of PD1 on the T cell, which then gets phosphorylated by the Src kinase, which then attracts phosphatase, which in turn gets phosphorylated by a Src kinase, and then the phosphatase is active.
Are the ITIMs generally found on the same molecule as the ITAMs?
No.
Yeah,
I'm
okay,
saying.
so they're
So that's right.
completely
So the
different.
ITAM, remember, is found in the zeta-zeta or one of the CD3s that we discussed.
Yeah.
Got that up front? Okay, but no. For instance, it's found on PD1, is an example I gave you. There are other ones as well, but no. To my knowledge, no, that's not the case.
So how is this important in the cancer?
Is it what, sir?
How is it important in the cancer?
Oh, how this is used in cancer immunotherapy? Is that what you're asking? Well, okay, I'll tell you a lot about that later on in the course, but there's a monoclonal antibody which can bind to PD1 and inhibit PD1. So you're inhibiting an inhibitor. You're not allowing the braking system to work. You know, if that were a car, instead of you braking going down a hill, you'll continue to accelerate going 100 miles per hour. You'll get to your destination faster, but you could crash, obviously. In this case, the monoclonal allows the immune system to no longer have a braking part to it. So you accelerate the immune response, hoping that you kill off the cancer cells. Downside is, because you may accelerate the immune system too much, you're going to have autoimmunity set in. Some problems, that's what happens. Some problems do arise because of that. That's how it works. Because then you inhibit the PD1, which has the ITIM on it, and then you inhibit the ability of phosphatases to be activated, so the system stays on. No way of turning it off. That's the question. Okay. But we're going to get more detail about that later. Okay. So more questions. Yeah.
Is that blue molecule? Is that the?
Is what molecule?
The blue molecule in the picture is
Blue molecule.
TIM?
No. That is showing you in this case, since there are no more red dots on the right there, that's showing an ITAM or some molecule which is tyrosine phosphorylated, which is now being dephosphorylated by the phosphatase. It's a pretty lousy diagram, so that's what that's showing. The reason we know that is because the diagram to the right no longer has those red dots on it. So. Okay. Other questions? So it's kind of a cool system because, again, what we know about the deactivation of the immune response is much more modest at this point than we know about the activation. This is one of the things we know for sure about how to deactivate it. Yeah? Allegra?
So would the PD-1 get secreted out? Because you said.
Now,
that-
PD-1 is not secreted. It's embedded in the cell, and it's on the cell membrane. Part of it sticks up on the cell membrane, out of the cell membrane, and then it binds to PDL1 on an APC. So it's not a secreted molecule. It's like the T cell receptor, which is not secreted. Then, okay, any other questions? Good, I got you interested in something here, so the negative aspect of immunity. Now, I threw this in for your edification about how TLRs signal, and I'm not going to go over this because it's just a bunch of names and we're running out of time anyway. But the reason I included it was not completely capricious. I had a reason to my madness. And the reason is I want to show you there's other signaling cascades that we do understand. And since we talked about TLRs almost at the beginning of the term, I just wanted to tell you we understand how TLRs signal as well. And this just shows you a whole bunch of molecules. A lot of tyrosine kinases are involved in this story. And ultimately, we had the same transcription factors being induced, nuclear factor kappa B, and also a MAP kinase cascade being involved. So very similar signaling cascade involved in TLRs. However, these transcription factors, although the same, bind to different genes. Because remember, we have euchromatin and heterochromatin, and in different cells, different molecules genes will be upregulated, even by the same transcription factors. So that's what's going on here. Okay? That was just to tell you that. Again, you won't be held responsible for this on exam, this slide and the one before, and I promise you that. I just want you to be aware of it. Not everything I tell you necessarily has to be tested on. Just want you to be aware that there are other signaling cascades as well. And again, this is the first one first one of the first molecules we discussed, and I wanted to understand, you know, what's going on. Okay. So that's the end of our signaling story. So let's get rid of that one. And now we're going to start a whole new topic. This is another one of my favorite topics. And I forgot to post this last night, so I did post it today, and I sent you an alert to that effect. Apologize, I forgot to do it. This is T-cell development in the thymus. And now we're going to learn the secret, the mystery. We're going to solve the mystery. Not of Edwin Drood. I don't know if anyone ever read that. All right, we're not going to solve that particular mystery, or the telltale heart, or the murders in the Rue Morgue, or the purloined letter. I'm in Dickens and Poe mode here. But we're going to learn how you learn to recognize self versus non-self. And you're going to leave this section of the course unsatisfied with my explanations. And the reason you're going to be unsatisfied with my explanations is there's lots of unknowns in this field. Lots of question marks. There's one more area, unlike blood pressure control and temperature control and diastolic volume and all that junk I teach in 115. There's a lot of things we do not know in immunology. That's what makes it so immensely interesting. As is 115, I don't mean to denigrate my own course, but this area really is a lot of question marks here. But I like it because it's mysterious. Remember, Charles Dickens died before he finished writing The Mystery of Edwin Drood. I have no idea what that has to do with this lecture, but so let's go. All right. Now I find out, by the way, if I take off my glasses, all of you are blurred out there, and I can't see if you're texting, I can't see if you're asleep, I can't see if you're bored, I can't see if you're interested. So it makes you all the same, which is kind of nice. But then when I put on my glasses, I see the truth. At any rate, what happens is we can do a lot of culturing here to figure out how we get T cells versus B cells. And a simple in vitro experiment is right from your book. So we can take stromal cells from the bone marrow. Stromal cells are fibroblasts, epithelial cells we take from the bone marrow. We grow them in culture. Being shown here. And then you can transfect the stromal cells to either express the Notch1 ligand, and it turns out these cells here, which might be stem cells, on the left and the right, have on them receptors for the Notch ligand. And to make a long story short, and it is a long story, maybe not even worth describing, but I find it kind of interesting, is that in culture, if you grow stem cells or progenitor cells and take the stromal cells and transfect them with the Notch1 ligand gene, so these cells now express Notch1, which will now bind to the stem cells or progenitor cells via the Notch receptor. If you have Notch1 ligand and then give growth factors in vitro, you'll get T cell phenotype. If the stromal cells have low or no Notch1 being expressed, the default phenotype will therefore be B cells. So I already told you way back near the beginning of the course during describing hematopoiesis what direction a particular cell will go into as far as its final differentiated state. Depends upon what cytokines they're exposed to and growth factors. But one more thing for B cells versus T cells, Notch1 ligand, if a particular stromal cell expresses that, you will have T cells. If it fails to express it, you'll have B cells. The Notch one itself is induced by a transcription factor called GATA3, which then begs the question, what upregulates GATA3? That depends upon which cytokine is present for the stroma cell either to have or not have the Notch expression. So we have to go back and back and back to get more and more explanation. In any case, it's a nice little, very simple story, and it is truly simple about how we can do either end up with T cell development or B cell development. And our story today is limited to T cells. We'll talk about B cells after the second exam at this point. Remember, I'm going to cover on the exam everything up to and including this Thursday's lecture. I've sent you an email. So yes, this will be on the second exam. And by the way, did you also get my email about when I'm having office hours? Don't come between 12:30 and 2 because I stupidly, very masochistically, scheduled to have an exam in physiology the day before I have an exam in this course. So I can't have you all come during the same office hours. So your office hours are 4:30 to 6 on Tuesday, Thursday, immediately after this course. That's the best I could do for you, okay? Without going crazy. Not to mention, I don't know when I'm going to get these all graded. All right. Now this is a review of the etiology of the T cell receptor, or development, ontology I should say really, of the T cell receptor. The beta chain is rearranged first. Beta chain is rearranged first. And once the beta chain is rearranged, you then get what's called a pre-T alpha chain expressed with it. At this point, TDT is downregulated. And this also sends a mitotic signal to the T cell, so you get a lot of proliferation. And then that proliferative stage ends. And then you get alpha chain splicing, and then eventually you get a mature T cell receptor with both the alpha and beta chains being expressed. But at this point, once you get both alpha and beta chains being expressed, your T cells are originally double positive. That means they express both CD4 and CD8. And what we're going to learn in this section is, first of all, how you go from double positive to single positive. How you go from DP, it's called. That's the jargon. DP, double positive, that means expresses both CD4 and CD8. And how you get to the single positive stage, whether you're CD4 or CD8. Almost no peripheral T cells are double positive for CD4 and CD8. We can do that by flow cytometry easily enough. There are infinitely small number of double positive T cells in the periphery. Yes, there's always some exceptions, but very, very few. And then we're also going to have to learn how this T cell, which we really shouldn't call a T cell yet because it hasn't graduated, it's called a thymocyte. Just like all of you are underclass people, instead of underclassmen, we don't use that term like that, so it's underclass people, that sounds bad. You don't have a degree yet, so you're not graduates. So T cell isn't called a T cell until it gets its diploma, and it doesn't get its diploma until it goes through selection in the thymus. So until it gets its diploma, we call it a thymocyte. Instead of an undergraduate, we call it a thymocyte. Okay. Now how we're going to get the undergraduate into getting its diploma, and then a nice dinner with their family, and then the abyss before them, because now we're going to do with the rest of your life, is we have a lot of steps to go through before we get that diploma, and the uncertainties, existential meaning of life confronts you. Okay. Now, this is a slide with a heck of a lot of information on it, and we're not going to be responsible for a lot of facts, which are interesting, but I don't want to belabor the facts. You notice there's 4 double negative stages. I'm not interested in you knowing that. And we have various molecules, either upregulated and downregulated, as we go through the 4 phases of the double negative stage. Double negative means we have neither CD4 nor CD8. We're going to skip that entirely. Which I did already, I suppose. And we're going to go to the phase where I'm not interested in all these phases. We're going to skip all that. So we're going to go, as you already know, I already said you get beta chain rearrangement first. Okay? And at this point, when you get beta chain more or less completely spliced, more or less, thymocyte now starts to express CD3. So we get the beta chain rearrangement, you can see D to J and then V, D, J. That's the point where we then get CD3 being expressed as well. So already, at this very early stage in the thymocyte's life, or development, we have the beta chain and starting the signaling apparatus, because we're now expressing CD3 as well as zeta, zeta, by the way. You can put that in your note if you want to. Zeta, zeta also is here at that time. Then we get the pre-T alpha, as I already explained in the previous slide. Then we get alpha splicing after we get proliferation. And at this point, once you have alpha and beta both expressed, you switch from the double negative to double positive. And make a long story short, then under a selection process, you either become CD4 or CD8, not really both. And if you survive all these various steps, you then leave the thymus and become a mature T cell out in the periphery, trying to find the antigen that you should be reactive against. So mature T cells then leave. So at this point, once you leave the thymus, you're no longer called a thymocyte, you're called a T cell. T cells here, thymocytes over here. All right, there's also a side step here, the gamma delta cells. Remember, we briefly talked about gamma delta cells. It's been shown in embryos of mice. The gamma delta phenotype is produced first, followed by the alpha beta. Gamma delta, I told you, might be able to recognize lipids amongst, as well as peptides. Gamma delta is produced first, presumably evolutionarily advantageous, because it seeds your intestinal tract, your respiratory tract, etc., and may act as sort of a bridge between the innate and adaptive immune responses, protecting you from a certain numbers of bacteria primarily, not viruses, but certain number of bacteria. So, but we're not going to talk any more about gamma delta cells in this course. They're quite interesting. Some people devote their entire research life to trying to figure out what they do and how they are produced. But we're going to skip them, and with all my apologies to people who study these types of cells. The far, far largest number of T cells we possess in our repertoire are alpha beta, and that's why I'm justifying just talking about the alpha beta. The far, far highest percentage of cells. T cells are alpha beta, so that's what we're going to be talking about. Right. Now, in order to make sure two things take place for a mature T cell, you want that mature T cell to have the following two characteristics. You want that T cell to recognize your own MHC molecule. Because if your T cell receptors can't bind at all to your own MHC molecules, they're not going to be able to bind to an APC and become activated. Remember, the T cell receptor recognizes two things simultaneously, dual recognition, the MHC plus the processed peptide. If the receptor can't bind to your own MHC molecules, it's useless. And the process by which the thymus looks at T cell receptors in which it selects those which can bind to your own MHC molecules, it's called positive selection. So a thymocyte must go through the process of positive selection, which means it's being tested to see if it can recognize its own MHC molecules on APCs. if a T cell receptor that a T cell just accidentally put together by splicing VDJ and VJs, beta, alpha respectively, chains, fails to bind to your own MHC, it's killed off. Something called death by neglect. Death by neglect. On the other hand, if that thymocyte binds too well to an MHC molecule and starts getting a signal to start being active, that's bad too, because you don't want a T cell to be reactive against your own MHC. You only want it to bind there. You don't want it to be activated by just your own MHC molecules or your own peptides, which are in the binding cleft of that MHC molecule. You want it to be able to bind, but you don't want it to be just activated by self molecules. There has to be a foreign peptide buried in there for the T cell to be activated. So if in the thymus, our thymocyte binds to some MHC molecule and starts getting activation signals, such as ZAP70 being activated, etc., that's not something you want to see turn out. Those cells, thymocytes, are killed, and that's called negative selection. And it turns out this whole process used to be called thymic education. But people thought that was a bad term because education should have a positive connotation, not a negative connotation. And it turns out over 96% of the thymocytes are killed in the thymus because they fail either positive or negative selection. So people decided they don't want to call it thymic education anymore, because I would hate to see 96% of you being killed off before you earn your bachelor's degree here at Tufts. We'll take your money. But we don't want to kill you off in the process. So they decided to call it thymic selection. So whatever term you want, it's fine. But they did change terms. I think that's an inside joke in immunology, I guess. Ha ha ha. But it's not called thymic education. Now, the dividing line between positive and negative selection is difficult to establish. And the people then thought, well, maybe there's something called the Goldilocks theorem or hypothesis. And the everyone read The Three Bears? Remember the porridge, it was too hot, too cold, or just right, et cetera, et cetera? Well, the idea is if you bind too tightly, that's not good. So you're killed off by negative selection. If you don't bind tightly enough, that's not good because you don't recognize your own MHC molecules. So you're killed off because you fail positive selection. And then you're left with the cells that are just right, thymocytes, which recognize self well enough to bind, but not to become activated, and that they actually therefore survive both positive and negative selection, exit the thymus gland with a degree, and become mature peripheral T cells. All right, so my goal now is to educate you about what positive and negative selection are and how they may work. No one really knows. So this is a great slide. I want you to look at it for a minute and a half. So please, I know I gave you these slides late, but I want you to look at it for a minute and a half and look what's going on in our selection process, okay? This is the cortex of the thymus up here. This is the medulla down here. Let me just give you some acronym interpretations. CTEC is not the airport in Seattle, although it is. This is called cortical thymic epithelial cells, CTECs. I guess CTEC, not CTEC. CTEC. In the medulla, we have MTECs, which I'm sure you can figure out what that means. Medullary thymic epithelial cells. And we also have bone marrow derived, I should say, dendritic cells. Double positive cells, double negative and double positive cells exist in the cortex. By the time you get into the medulla, we have single positive going on. Okay, so I want you to look over this slide and look at all the intricate detail there, because this summarizes everything you really should understand about positive negative selection. And then we're going to do a lot of experiments to prove the things that are going on in the thymus. So I want you to look this over. So do your homework now. Look over this particular slide. All right, look it over. Don't stretch and go on. Look it over. All right. By the way, I like to interact with classes more than I can in this room. I like to usually go up and down the aisles, but I cannot do that here without falling over and killing myself, because these desks are so very close together. So I'm forced to stay in the front. I'd rather perambulate around the class. Can't do that. All right, I'm going to help you through this, of course. Ta-da! What are APCs? Give me the APCs. Give me the APCs. Quickly, tell me what the 3 APCs I've told you about. Dendritic cells, number 2. Number 3. Well, turns out I lied to you. There's another type of APC, which you can find in the thymus gland. The third type of APC is neither a B cell, nor a dendritic cell, nor a macrophage. It's an epithelial cell. So it turns out the C-tex and the M-tex are APCs. In fact, very, very good APCs. They express both class 1 and class 2, one of the few epithelial cells in our bodies which express both class 1 and class 2. Most peripheral epithelial cells outside the thymus only express class 1. But like most things in immunology, here's an exception. In the medulla, cortical and medullary epithelial cells express both class 1 and class 2 and can process peptides endogenously and exogenously. Okay, so what happens is, here's our thymocyte double positive entity. We've gone through our 4 stages of double negative, which we're not going to be responsible for. And it turns out, in the cortex, some of these cells can't bind at all. They don't recognize anything that they can bind to. So most of these cells die by neglect. And you can see the numbers, 90 to 96% of the thymocytes in the cortex die by neglect. They have T cell receptors which cannot recognize any of your MHC molecules, which are all displayed by the cortical thymic epithelial cells, class 1 and class 2. Yep.
Are they actively killed, or do they?
Yes, they're actively killed. How they're actively killed is by apoptosis, and I'm glad you asked. There's another step here I'm now going to interpolate here. It turns out that if a thymocyte binds to C-tech just right, it upregulates a molecule called BCL2. As long as you ask, it's a good little point maybe to know. And BCL2 is an anti-apoptotic molecule. It inhibits caspases and prevents, therefore, destruction of that particular thymocyte. And by the way, some cancers have an overexpression of BCL2, and that means you can't undergo apoptosis when you should, and therefore these cells can become malignant. But that's another story. In this case, if the cell doesn't bind, it does not express BCL2, the cell gets a signal to activate the caspase cascade, the apoptosis cascade, and ends up destroying the cell. Okay, does that answer your question? Okay. That's death by neglect. You got to love that. That should be a title of a Halloween movie. All right, so I should have done this tomorrow. Death by neglect. That's a cool thing. All right. On the other hand, if it binds to the C-tech and gets just the right signal, it's then allowed to go into the medulla to undergo further processing. On the other hand, if it binds too tightly to the C-tech, it undergoes negative selection because in this case, you end up signaling and a BCL2 is downregulated and you end up leading to apoptosis, so the cells are killed off. Killed off just right. Mostly, though, positive selection occurs in the cortex, and mostly negative selection occurs in the thymus. In the medulla, and you're going to find out why that's the case. Most positive selection occurs in the cortex. Most negative selection occurs in the medulla. However, there is negative selection occurring in the cortex as well. And mostly what we're selecting for in the cortex is our ability to recognize self-MHC or not recognize self-MHC molecules. Mostly that's what's going on in the cortex. Okay, everybody with me so far then? Okay. Then, as we migrate into the medulla, we become single positive. Everything's double positive at this point. This is all double positive. Then we move down here, and we're single positive. And then there's this miracle molecule. And it truly is a miracle molecule, which is acronymed AIRE. And AIRE, and I have a slide about 6 or 7 or 8 slides from now, maybe 50, I forgot, where this is all typed out for you. Plus I have a sample question about AIRE, because I love this molecule. Which means if I have a sample question with an answer about AIRE, it's not going to be on the test, because I already given you the sample question with the answer, but still a cool molecule, even if you're not going to be tested directly on it. AIRE is mostly expressed by MTECs, medullary thymic epithelial cells. And what it does is AIRE binds to regions of the genome which are histone regions which are such that they normally would not bind RNA polymerase II. They normally would not bind RNA polymerase II. And AIRE binds to those parts of the genome, opens them up, attracts transcription factors and RNA polymerase II. So AIRE is a master switch which leads to the expression of almost every gene in your genome in the medulla of your thymus. So for example, AIRE will cause the medullary thymic epithelial cells to produce insulin. Now normally we only think insulin being produced by our beta cells in our pancreas. But AIRE upregulates a whole range of genes in our genome. Why would it do that? Because you want to become tolerized to self molecules. And in order to become tolerized to self molecules, you better express those molecules during thymic selection. And it turns out, not in the cortex, but in the medulla, we have AIRE in these MTEC cells. That leads to the expression of almost all the gene products in our genome, not all, but many of the gene products in our genome, including insulin, etc. And you become tolerized to that. So you don't have T cell receptors in the periphery being reactive against insulin. Now sometimes that does happen, and you can end up with, unfortunately, type 1 diabetes. But most of the time, people express insulin and then AIRE because of AIRE and become tolerized to it. So in the medulla, we have AIRE. We express, for instance, insulin both with class 1 and class 2 on our MTEC because the insulin is being produced by the MTEC, and therefore that can be class 1, plus it's secreted and then gets endocytosed, and then for its class 2. So insulin or any other molecule is expressed both with class 1 and class 2 in the medulla. And turns out dendritic cells, bone marrow derived dendritic cells, don't have air, but they can take the products of air from the MTEC and express them mostly with class 2 because they're not synthesizing the molecule, so they just have it the exogenous pathway with class 2. All right? So go through this whole process again now that I've told you all these secrets of this slide, which has all these little details, which are really summarize everything I'm going to tell you about positive and negative selection. And after you master this slide, I'm going to go back and give you a whole series of experiments which show you that positive and negative selection actually take place. There's been like a lot of experiments, but before you do that, I want you to master this slide. So go ahead and look at it. Don't look at me, don't look at each other, look at the slide, okay?
What happens to the BCL2 in the medulla if those,
It gets deactivated.
it gets,
That, like everything else, it's upregulation and downregulation. The fluctuation. And I know there's a heck of a lot of acronyms, but you really should memorize MTech and CTech. And I want you to memorize what that represents.
What's the advantage of having eight long medulla?
That's a good question.
Doesn't necessarily like positive or
Well, this way, remember, the negative selection can't be negatively selected in the cortex against all of your different cell molecules, because now it's expressed in the cortex. So maybe it's better off that it's single positive, because this occurs in the single positive stage. This negative selection occurs in the single positive stage. That's the best thing. Good question. All right, everyone feel comfortable with positive and negative selection? Where does most negative selection occur? In the cortex. No, where does it mostly occur? Because why? There's one good reason. Why? Because what's expressed there but not in the cortex? So therefore you get a whole outpouring of self-antigens being produced, which you can become tolerized to. All right, where does most positive selection occur? In the cortex or medulla? Cortex, okay? However, can we also have negative selection? Presumably, yes, against self-MHC molecules, and also some self molecules expressed there as well. And by the way, self-MHC molecules, you may become tolerized or negatively selected against them either intact, or the self-MHC molecules get shed, then endocytosed, so you can actually be expressed in both class 1 and class 2 as a result. So that's another way of just looking at their subproducts that you may become recognized for the TCR. So that's something to think about. All right. This is just a cheap, dirty cartoon showing you the anatomy of the thymus, showing you where it is. And remember, as you age, the thymus involutes, shrinks. This dramatically occurs at puberty. And for every decade of life after that, it shrinks more. And there's a condition as you age. Immune senescence. As you get older, your immune system becomes less and less potent. Like everything, as you get older, things start breaking down, and therefore older people are more susceptible to infectious disease than younger people. And that's due in part, but not exclusively, to the fact that the thymus starts to involute from puberty onwards. So all of you in this room are already on the downward slope as far as your immune system goes. All of you are having, starting and are well on the way to having your thymus glands involute, become smaller. The only cure for that is death. All right, so let's look at positive selection first. All right, we're going to have transgenic mice, which means in their zygotic stage, we put in a gene which codes for an already spliced together T cell receptor. This is an interesting trick. The transgenic receptors we did to these mice are class 1 restricted. It turns out if you make a mouse transgenic for one particular T cell receptor, that stops splicing of the beta and alpha chains. So the only T cell receptor this poor mouse is going to have is the one that you gave it transgenically. The only one it's going to have. It's a pretty awful thing to think about. But nonetheless, that's what's done. That's because of allelic exclusion. And okay, look at this. Get the secret here, what's going on? It's not a lot of data here, not a lot of variables going on here, but see if there's enough that should make you think for around 28 seconds. See what's going on here. I already told you enough. It's a transgenic mouse. The only TCR virtually it's going to produce is the one that it was given preformed. So by allelic exclusion, it won't make it anymore. And we put this transgene in a mouse, which is either MHC on the left or the MHC on the right. And everything else, there's enough information here for you to figure out. We're going to have a lot of these type of cartoons, a lot of fun with these experiments, a lot of transgenic experiments today, on Thursday. All right. Yeah, I'm giving you hints. Okay. Why do we get cells on the left? Why don't we get any cells on the right? Anyone want to volunteer? I could give the answer, but I'm too lazy. Come on, some brave soul out there. Go ahead. I feel like you want to raise your hand.
Me?
Yeah. I misinterpreted your body language, okay. Yeah.
Because the mouse is restricted to MHCA.
For haplotype A on the left, right. And the transgene, what kind of haplotype does the transgene recognize? It's class 1 restricted, but what type of haplotype does it recognize? Type what? A, right? And therefore, will it undergo positive selection? Will it be able to bind to an A haplotype MHC molecule? Yes. However, on the right, the transgene is still an A transgene, but what is my mouse? It's a what type of haplotype mouse? B. So there's it's going to fail what type of selection here? It won't be able to bind to the B haplotype, so therefore it's not going to bind at all, so it's going to fail what? Positive selection, because it failed to upregulate what molecule? BCL2 amongst others, and therefore you fail positive selection. And this poor mouse, not only is it doomed because it only has one type of TCR because of allelic exclusion, it doesn't have anything because it failed positive selection. 100% of its cells fail. It's like if I gave the second test and the average was 20 with a range of 10 on either side of that, that means I failed with you. All right? So this is what happened to these thymocytes. I had a really, really hard professor. That individual failed every one of these thymocytes. You can imagine what the teaching evaluations would be for this thymus. But thymus is going to have to worry about tenure and labor. All right. Okay. I guess I don't either because I'm retiring. So. All right. Do this one. We're going to get more and more complicated experiments. We're starting simple here. This is fun. This is why I like this. We build up our complexity of experiments. What can you infer from this? Transgene is what restriction is this? Transgene class, and this one is class what? Therefore, can a TCR, which is class 1 restricted, bind to a class 2 molecule? Can a class 2 bind to class 1? So therefore, the only thing you're going to get is class 1 restricted and class 1 because it will bind to class 1 molecules. Presumably it recognizes the MHC molecules of this particular mouse. And it turns out if you bind to class 1, you know already, if you're going to go from double positive to single positive, if you bind to class 1, what's your educated guess? Which CD are you going to prefer to keep expressing? CD4 or CD8? CD8. You're going to come single positive CD8 because just by accident, your T cell receptor bound to a class 1 MHC molecule. On the other hand, if you made a TCR just by accident, which was class 2 restricted, you go from double positive to single positive, but because you're class 2 restricted and you're undergoing positive selection, which of the two CDs will you retain? The CD4, which is class 2 restricted, or the CD8, which is class 1 restricted? Which one would you retain? CD what? 4. So that's why in this particular mouse, the only thing it's going to have in its periphery is CD4. The only thing it's going to have in its periphery is CD8. All right. This is more complicated. We're going to get progressively more complicated. Read these 4 squares, the first 3 anyway. I don't care as much about the last one, but read these 4 rectangles or squares and see if you can figure out what's going on experimentally and what the results tell you. Again, I could do that for you quickly, but I don't want to. Actually, do look at fourth one as well. All right, if you want to discuss this with your neighbors, I'd like to hear some conversation going on here. There's some stories that you, or some moral to the stories that you should conclude from this.
And the third one, it's still like, they don't have the MHC, and they everything is the class 2, but the class 2 has the artificially put in, so they can still express.
By the way, before we have fun figuring this out, what kind of assay would you use to see if you have both CD8 and CD4, or only CD8, or only what kind of assay we've used many times this semester would you use? Flow cytometry to see if you have, okay, all right. So, okay, if you have normal MHC class 2 expression, presumably you also have normal class 1 expression, so you get both CD8 and CD4 cells maturing. if I have a knockout, so it can't produce MHC class 2, what's the only phenotype, single positive phenotype it will therefore have?
That's one.
Right, class 1 CD8. So the only cells this animal will have in its periphery are CD8 cells of various specificities, but the only restriction will be for class 1. However, due to the brilliance of molecular biologists, I can have a knockout mouse for class 2, but then I can get class 2 only to be expressed by cortical thymic epithelial cells. And that shows what? Cortical thymic, if I only have it on cortical thymic epithelial cells, cortical thymic epithelial cells can affect or bring about positive selection. Because I selectively induce class 2 only on the cortical epithelium. That shows there may be other cells leading to positive selection, but it certainly shows you the CTEX. Those are the only cells I'm allowing to express class 2. Obviously can bring about positive selection. Other cells might do it. We know that isn't the case, but certainly that says they're capable of inducing positive selection. Other cells might as well, but I'm telling you that's not the case. But the CTEX, okay? And finally, what's the last rectangle tell us? We're expressing class 2 on our cortical thymic epithelial cells, but we mutated the class 2 so it can't interact with CD4, and if CD4 can't bind at all to class 2, that adhesion, that extra adhesion doesn't take place, so you'll fail positive selection. So that extra adhesion between the CD4 and class 2 is necessary to get positive selection. So this is a nice, cute slide, which I won't ask on the test because I'm giving you, again, a sample question with this on it as well. So, okay, figure this one out. We're going to get progressively more complicated. This is now putting together all the jargon and all the stuff you learned about the MHC, haplotypes, etc. Just think of all the unbelievable information that you know. Just think of all the words you learned in this course. You can't possibly use it on the I already told you what transgenic mouse is. That means it can only produce that TCR because of allelic exclusion. It really can't produce any others, or not many others. Again, talk down with your friends. And then big reveal in a second. see if we can figure this out. Okay, what's the restriction for this TC receptor that we're going to transgenically transfer to these two different mouse strains? What is the restriction of this particular transgene? What's the restriction? What's the restriction? Class 1, what's the haplotype? Okay, what's the specificity? Again, these are terms that you have to know for the second test. So what's the specificity for this particular TCR? All right. Let's say we had a bird and the bird's name was Enza. It came in that window. What came in the window? Influenza. That's how that disease got its name. All right, so influenza. All right. So here is the TCR. What's its restriction? Type, class, not U class. What class? MHC? 1. And what's its haplotype? Okay, and what's its specificity? Something on influenza. Okay, let's say hemoglobin, just sake of argument. And then all right, so what happened here? Someone tell me. Yeah, we're showing positive selection. We have the transgene, which is haplotype K restricted, and the poor mouse is D haplotype. Will you be able to bind successfully in the cortex? No, so you'll die by death by neglect. You'll fail positive selection. So this poor animal has no thymocytes left, but this animal is perfectly fine because it's the K haplotype. So therefore, his ability to bind is sustained. So we upregulate BCL2, and we're ready to go. And that leads us to a Nobel Prize experiment. The Nobel Prize experiment is done by Zinkernagel et al. And this really revolutionized our understanding way back in the 20th century of understanding of positive negative selection.
On the D mouse, why is there a plus then in the first?
Because double positive hasn't undergone selection's just taking place. There's lots of cells being produced, thymocytes, so you're going to have double positive. And then finally, when you're single positive, nothing left because everything's failed. All right, so we're going to do this next time. LCMD we talked about before, so look at the control and look at the experiment. Before you go, I just want you to note, look at all the things that were done to this poor mouse, and just think of the experimenters who had to do all these procedures. That's why they got a Nobel Prize. It wasn't a cartoon then. All right, there was a lot of steps involved here. So I'll see you all on Thursday.
I see that email, thank you.
All right, yeah. I thought no, it's SGA. Well, it's SGA now. It's like our brand becomes very proprietary. No, it's pretty one who violate any illegal issues. All right, I'll talk about it. Okay. I'm busy these next 2 weeks.